Good day. On this session, I'll be attempting Life Science Paper 2 from September 2022. So it was a trial exam which was written in the Limpopo province. So I'll be attempting all the questions and hopefully I'll be able to finish in the allocated 2.5 hours. Without wasting any time, let's go straight to question number 1. Question number 1, it's those 10 multiple choice questions. So the most important thing when you're given a multiple choice question is to read the paragraph before you look at the given options. We are being told that the sequence of the bases has got the following sequence: UCA, CGA, ACC, GCU, AAC. So they're asking us that given sequence, it could represent what? Option A, it can be a gene that codes for protein. Remember that we know it's an RNA because we've got uracil. So now you've got uracil, we know it's an RNA. And then also we know that for us to form amino acids, we're going to form that, and that. So meaning this will be a code that will give us amino acid. So in other words, the most obvious option from the given one will be an mRNA strand that codes for 5 amino acids. That would be 5 amino acids. It can't be a DNA. DNA audits out. So the other thing that you can do is to take out the obvious answers out. It can't be a DNA because there's a uracil there. A chain of 5 amino acids. Remember, we need to also know what the tRNA will be. So we can take that out. Also, it can't be one protein because we need to get those 5 amino acids. So it can't be one protein. So when you can get 5, you'll get more than one amino acid, more than one protein here. So because I'm getting more than one protein, we can't have one protein because we'll get more than one. So that one is out. So meaning the obvious one will be option D, which means it will be an mRNA that will code for 5 amino acid. So let's move on. We've been told that we have a male whom is homozygous for blood group A. So let's just do the male. So the male is homozygous for blood group A. So meaning if it's homozygous, it will be 2 full letter A's. That's homozygous for blood type A. And a female with blood group B have a children. So that male, just call that SM. So we have that as our male, and then our female is blood group B. So we're not sure whether it will be BB or BO. Can be BB or BO. So those can be our different options that we can get there. We can have BB or BO. So now they are asking us which one of the following genotype is possible for the offspring. So which one will be possible for offspring? We know we've got a male who is homozygous for blood group A, and then we've got a female who will be homozygous for, not homozygous, who will have a blood group B. So the most common allele options we're going to get here is that we're going to get allele option, which will be A and B. So we can have one allele which will have A and B based on the possible combinations. We can get the first allele, which is A and B. Again, we can get another A and B based on that. So we can get also another A and B. So if we were using BO, for example, if we're using BO, it would have been A and AO as other possible option. So the possible options you can get from female being BB or BO with a father of homozygous AA. Those are the 3 possible options you can get from those 2 parents there. Those are the 3 possible allele options. So let's go to our options. We don't have double AA, we only have AB. So AA will be out because we don't have double AA. So AA will be out. You can't get allele AA there. we can only get A, B, or AO. We've got A, remember, if it's not there, we know that that will be O. So basically this is A and O, which is possible based on what we got on the three different genotypes here. So A and O is possible, that will be number B. So letter B. Here we've got OO, this will be OO. So OO, no, it's not possible, OO there. And then this one would be BO. Do I have BO forming on the kids? These are the three kids we're gonna form. There is no BO that is formed there. So it's also out, which means our answer will be letter B. So the three possible allele options we're gonna get will be AB and also AB and also AO. So AO is letter B that is being formed. The other ones are not possible for the kids. So letter B would be our answer. So now let's go to, again, we're still doing genetics. So we're being told that color blindness is a sex-linked trait, so meaning it will be on the X chromosome. It's caused by a recessive allele on X chromosome. Okay. The pedigree diagram of shows inheritance of color blindness. Okay. So square light, that's a normal male. So this is our normal male there. We've got our normal male there, and then the female has got condition here. So we've got a normal male. So a normal male will look like this now. So it's an recessive allele. So for the male to get the condition, they must have a small letter allele. So because it's color blindness, I'm going to use a small letter C for this one. So because they don't have a condition, I'll use a big C to show that they are normal. So my male is normal there. My female has the condition. And remember, if it's a recessive condition, you need to get two small C's for them to get the condition. Because if they've got one, then that will be an carrier. But because they've got the condition, we'll put two small C's to show that the female has got the condition. So this is the male who's normal, this one here, and the female with the condition, this one here, will be that one based on our genotypes. So now from these two parents, we are having a male who's having a condition. So the male who will have a condition will be this one. So this male that will form here will be X and then small c there and Y. That's the male who will have the condition. That male here is the one that will have a condition. The other possible combinations we're going to form will be that and that will be a carrier. And then we're also going to have a female also. We'll have that and that, which will be twice. So it will be that and that also, that and that. So we're going to have two females who will have big C there, and then also small c. So those will be the two females who will form. And then lastly, we'll also have two males who will have the condition. So the two males who will have the condition will be like that. So we're going to have two normal females now. Just call it 2NF. That's normal females. And then also two males who are sick. Just call them CM, colorblind males now. So we're going to have 2:2 ratio here as the possible combination. So now being told that person 1 and person 2 have a child. Who's person 1 now? Okay, now this is a bit confusing. Who's person 1? Oh, this one. Yeah, that there. So the person 1 is this one here, and then person 2 will be this one. Okay, that's fine. So person 1 and person 2 have a chance, have a child. What is the chance that the child will be colorblind? If we have got those two parents having kids, 50% chance of them having a colorblind son will be possible for those ones there. And then we have now this female here. Remember, this female here will be this normal female, but she'll be a carrier. So when you have got person number 1 and 2, these two here, so the female we know will be a carrier based on this combination here. The mother will be a carrier, meaning they'll have one big C, and then the other C will be small. We call them carriers. They are carrier for color blind. That's the mother, this one here. The father here is normal. So if the father is normal, meaning the C will be a big C now. So a C will be the big C, and then the Y will be that based on the son or the man being normal. So it'll be a big C. So now the possible combinations when I get will be big C and big C, and then also big C and that Y there. So we're going to have also small c and small c, that will be normal female. So we're going to have that also small c and y. So we're going to have one boy who will be x, c, and then y. So there's only one child out of four. One child out of four who will have the condition. The other three are gonna be normal because they've got a big C in them. Because this one that we formed first, this one and this one here, they're forming. Just trying to erase that so that I can show you guys again so that I don't do a lot of errors. This one here and this one here, they've got a big C in them. So that means they'll be normal. So it'll be C, big C, and also X with also big C. So meaning they'll be normal because they've got big C's in them. Same. The second one, boy that will form here will have a big C from the mother, meaning the boy will be normal. Then we're gonna have this, and forming with this X, it will be the small c and big C. As long as you've got a big C, meaning they'll be normal. So it's only one son who will come with a small c, meaning 1 out of 4 possibility of them having the condition. And we know that 1 out over 4 will be 25%. So 1 over 4 will be 25%. So I mean the possible answer will be 1 over 4. So here was for these two parents here, which they didn't even ask for. I don't know why I took a lot of time for that, but they're asking for these two. So just quickly go straight to the question. Don't be like me, we're gonna waste a lot of time doing the different they didn't even ask us to do. So the possible answer there will be 1 over 4, which is 25%. And then now we're going to a different topic, which is evolution now. So we're given 4 limbs of vertebrates. So they're given 4 limbs of vertebrates. So this one is about knowing different type of evolutions. So we're being asked the type of evolution or the type of evidence for evolution which is shown there is what. So basically they want us to find to know the different evidence of evolution. So they want us to know the different forms of evolution. So the type of evidence of evolution shown here will be that's modification by descent, because we've got different 4 limbs which are being modified. So that's modification by descent. So they're being modified from different limbs. So that will be the obvious out there. Then let's go to number 1.1.5. We'll go back to genetics again. So we've been given chromosomes now. We know that a normal human being will have 46 chromosomes, and then remember they're in pairs. So each must be in pairs. Oh yeah, we've got 21 there in triplets. Remember, we've got trisomy 21. So meaning the child must have Down syndrome based on trisomy 21. So we're being asked which statement will go with the karyotype that's given to us. So now we're given different options there. So remember, we know whenever we are dealing with trisomy 21, there's always non-disjunction at anaphase. So we know that non-disjunction must occur. So the good thing is that all given options are having something about non-disjunction. But you know that for us to have trisomy 21, non-disjunction must occur. So option number A, yes, we've got a non-disjunction occurring. B, no. So audit out B. Before you even read other things, B is out, because for us to form trisomy 21, we must have non-disjunction. Same as C, non-disjunction must occur. So it will be out also C. So meaning possible answers will be A or D. Now we go and check our karyotype. Remember, if it's XX, that's a female. XY is a male. We are having a karyotype of XY, meaning the person must be a male. And option A is a male, because D is saying it's a female. It can't be a female because it's XY. So meaning it must be a male. Meaning our answer would be letter A. It's a lovely question. Now we go to 1.1.6. We're being told that changes to the genetic material of organism can only be inherited by the next generation if they. So now we need to know how a change can occur in a different or in a organism. So they're asking us a change can occur in a genetic material only if it's inherited by the next generation. If they must occur in the cells which are causing reproduction. So the cells which are causing reproduction, so it must occur in our sperm cells or our ovaries. So it will be this one. Those changes must occur in the cells which are undergoing reproduction. Can be somatic, and then the other ones are different topics. So it's only gonna occur if it occurs in the cells which are undergoing reproduction, meaning letter C will be our answer. 1.1.7. Okay, below we have been given a list of fossils which are found in South Africa. So we're given 4 different options: Downchild, Littlefoot, Karabo, and Misplaced. Here right now in evolution, so it's all about knowing about different fossils and which genus they are from, who discovered them, because it's very important to know who discovered what. If they ask about Arthropithecus sediba, you must know that Liebega is the one who found them. Now they're asking us which of the fossils above are classified under Australopithecus. So we need to know who is classified under Australopithecus. So now, first thing that advice is that you need to go and tick all the ones that you know are under Australopithecus. So you must go and tick all we know are under Australopithecus. Downchild, we know that's under Australopithecus, same as Littlefoot. It's also under Australopithecus, same as Karabo, same as Misplaced. Yeah, they're all under Australopithecus. Oh, okay, that's good. Then let's check our options now. We've got letter A to D. All 4 are under Australopithecus, meaning B is the one that is having all 4 of them. So B is having all 4 of them. So that should be your answer because it has got all 4 of them because they're all under Australopithecus. Wow, that was a nice question. Okay. So let's go to 1.1.8. Now we're going to DNA. So DNA from cells of an animal was analyzed in a lab. The results are shown below. So they're analyzing DNA in a lab. Then they found those results. Remember, DNA must have guanine, thymine, cytosine, and also adenine. So I'm given guanine there, and the value of guanine is 26.3. Okay. Then we're given X, Y, and Z. So now we need to go and use the given values or percentages to figure out who's going to be cytosine, who's going to be adenine, who's going to be thymine. Because X, Y, and Z are those 3 other bases which they didn't give us. So now remember, guanine in DNA, guanine, same as RNA, guanine will always go with cytosine, and then we know in DNA, adenine will go with what we call thymine. That's in DNA, and we're in DNA here, so that's fine. So now the values of guanine and cytosine, they tend to be close to each other, same as adenine and also thymine. Their values are going to be close to each other. So with that being said, the value of guanine is bound to be close to the value of cytosine. So you must be thinking about such things already. Guanine must be close to cytosine in that way. So we know we're given adenine, so we're given guanine as that value. So now we must ask ourselves now, if that value is our guanine there, therefore the one that is closest to our guanine here is what? Cytosine, which is this one. 26.3 and 26.7 are close to each other, meaning Y must be cytosine because the values must be close to each other. If that is our J, therefore our C must be that one. So meaning this one already I know that's our cytosine. So Y must be cytosine. So I go to my option. Wherever I see Y, it must be cytosine. It's only letter C which is having Y as cytosine. That's fine. And another 2, adenine and also thymine, must be close to each other. Meaning if X is adenine, Z must be thymine. So let's go and check now. X must be adenine. Yeah. X adenine, that's C there. X is adenine, and then Z must be thymine. that is that. So meaning that is our correct combination. Also, X could have been cytosine, which would be letter A and D, but Y must be cytosine. It's only this one which is having Y as cytosine. The other ones don't have Y as cytosine, so the other ones will be obvious out, meaning letter C will be our answer based on J and C must be close to each other, same as A and T in DNA. So whenever they give you guys values, compare the different values given, and then those two must be close to each other. And then we go to 1.1.9. Okay, now we're going to genetics. When a red shorthorn cattle are crossed with white shorn cattle, the offspring have both red and white fur. That's co-dominant. So whenever you've got two different colors in one person, that is what? Co-dominant. So if, let's say, the red and white kettles were forming pink, if they're forming a pink color, that would have been incomplete dominance. And then if they form both colors in one person, it's what we call co-dominance. Incomplete would be if they form a different color, for example, pink, or they form a green color, that's incomplete. But because now we have both colors, we call it co-dominance. So that would be letter D. Then 1.1.10, again, we're still in genetics. The diagram shows genotypes of two parent plants and 3 of the offspring. Okay, we've got 3 of the offspring. So plant 1 is heterozygous, big R, small r, same as plant 2, big R, small r. Plant RR is homozygous for R. This one is heterozygous. That one is also homozygous but recessive. So we've got 3 that's homozygous dominant, 4 that's heterozygous, 5 is homozygous recessive for the trait they're investigating. So the question is, which of the plants have the same phenotype? Which of the plants have the same phenotype? So remember, if we're saying R, big R is for red. Let's say if we're saying big R is for red, so meaning 3 and 4 are going to be red, because that would be big R for red. We can say small r, maybe let's say it's white color, for example, since they didn't mention anything about colors. So just assume that small r is white color. So if big R is red, therefore 3 and 4 have got big R in them, meaning they must be red. So they must be the same phenotype, 3 and 4. 5 would be white, because that would be that recessive condition. So 3 and 4 must have same color in them. And then we know that 3 and 4 have the same color in them. And then also 1 and 2 will also be red, because they've got a big R in them. So 1 and 2 are also red, same as 3 and 4. So 1, 2, 3, 4 are going to be same colors. Only 5 will be different. So meaning 1, 2, 3, 4 will be same color. Only 5 will be different. That will be letter B. So that was our lovely multiple choice questions. And then now let's go to biological terms, which will be more about definitions. So 1.2.1, a variable that is changed or manipulated during an investigation, that's what we call an independent variable. So that is what we call an independent variable. So it's what we call an independent variable. So that's what we call an independent variable. So that's what you can change or manipulate during the investigation. That's an independent variable. Independent variable. Then the other one that will be dependent on this one will be the dependent variable. So independent we can change. For example, it can be time, it can be anything on your x-axis that will be changed all the time. Because I can go from 2 seconds, 10 seconds, so I'm changing it all the time. So that will be the independent variable. 1.2.2, the law that describes how alleles are separated from one another during the formation of gametes. We call that law of segregation. That's what we call law of segregation. So it's law of segregation because we've got different laws. We've got law of dominance, and those are the laws that some of them we found after Mendel was doing his investigation. So that's law of segregation, where they're separate from each other. We call that law of segregation. law of segregation there, and then that will be our law. And then now a genotype consisting of different alleles. Remember, different. If you've got the same allele, for example, big R, we call that homozygous. But if you've got big R, small r, we call you heterozygous. So that's heterozygous. Those ones who are having big R and small r, for example, we call those heterozygous. They've got different alleles in them. Can be big, small, big B and small B, for example. It can be big R, small r. It can be big K, small k. Those will be different. We call them heterozygous. The complete disappearance of a species on Earth. That's like dinosaurs. We call them they are no longer available in this Earth, so they are extinct. But the complete disappearance, we call it extinction. That's extinction. But if an organism is no longer present, we say that it's extinct. But because the whole process is called extinction. The splitting of cytoplasm during cell division. That's, remember, after telophase day, we call that cytokinesis. That's when the cell will split into two different cells so that we can form whatever we want to form, like one good cell that the cell will want to replicate. That's cytokinesis. That's after telophase. So an opening on hominid skulls through which the spinal cord passes. Remember, it goes forward and back. That's foramen magnum. So always use that magnum ice cream that people have. People always say that sometimes some people maybe they are crazy or they're acting weird, and that's not the case. The reason why people are being mean or being dramatic is because they eat magnum. So we call that foramen magnum, which is a lovely ice cream, by the way. You guys must do all your exams, then your parents can reward you with foramen magnum, not foramen magnum, with magnum ice cream. So the one that is there on us as human being and also apes is called foramen magnum. For us, it can be forward. For other ones, can be backwards, depending on how you're moving. If you are like us who stand tall, it can go backwards. For those ones who are moving like the typical apes, it moves forward also or backwards. The region of a chromosome where the two chromatids are attached to each other, that's a centromere. We call that a centromere. It holds the two chromosomes together. That's a centromere. It holds the two chromosomes together. That's a centromere. Okay, so that will cover our biological terms. So we can quickly move on to next question. 1.3 now, it's matching A and B. Column 1 and column 2 can have A only, B only, both A and B, or none. So 1.3.1, column 1, genetic disorder linked to sex chromosome. So we need to know our sex-linked disorders here. So in hemophilia, we know it's a sex-linked disorder. Single cell anemia, no, not. So meaning our possible answer would be A only, because hemophilia, it is a sex-linked disorder. So it's going to be A only, because single cell is not a sex-linked disorder. So it will be A only there. The evidence used to support out of Africa hypothesis by tracing maternal lineage. So tracing maternal lineage, remember, we use the mitochondrial DNA for maternal lineage. If you want to trace the paternal one, we'll use the Y chromosome. But maternal one, because remember, we can't use the maternal Y chromosome for maternal because mothers don't have Y chromosome. So whenever we're doing maternal one, we only use mitochondrial DNA. You can't use Y chromosome for mothers because mothers don't have Y chromosome. So we need to be A only. We only use mitochondrial DNA for mothers because they don't have Y chromosome. But for fathers, we can use Y chromosome. Examples of reproductive isolating mechanism. So we need to know all those different mechanisms, such as breeding at different times of year, which we know is part of them, adapting to different pollinators, yes. We also have things like mechanical behaviors, infertility as well. So these two are part of isolating mechanisms, so meaning the answer will be both. So you're gonna have both A and B. It's gonna be both A and B. So there are like more than 4 of them, which will be isolating mechanisms. So it's going to be both A and B for that one. So that'll be that. So 1.4 now, the diagram shows a process that occurs during meiosis. Remember, that's crossing over. That's crossing over, and we know that it occurs in prophase 1. So they're asking us for the name of the process that is showing on the diagram. That's crossing over. Remember, it is occurring in prophase 1. That's where the chromosomes are crossing each other. We call that crossing over. So that's crossing over. That's where we have got two chromosomes which are moving from one to the other. That's crossing over. And then the phase in which meiosis occurs in that, remember, that's prophase 1. That's going to be prophase 1. Prophase 1. That's going to be prophase 1. That's prophase 1. Prophase 1. Prophase 1. Identify the part marked Y. Yeah, wherever the two are meeting, we call that chiasmata. We call that chiasmata. Some can call it chiasma, or they can call it chiasmata. That's the part where the chromosomes meet. We call it chiasmata. Or you can call it chiasma or chiasmata or chiasma or chiasmata. Either way is fine. You can call it chiasmata or chiasma. Either way it's fine. Now describe the process mentioned in 1.4.1. That's crossing over. So here, first of all, the homologous chromosomes, they pair up now. So we've got the homologous chromosomes pairing up. So I'll repeat again. The first point will be the homologous chromosomes will pair up now. And then we know that each chromosome will have two chromatids. And then from there onwards, the chromatids will overlap or cross over from each other. And then therefore we're going to have genetic material being exchanged from each other. Then the new one that will form will have a combination of the two different traits. So the first thing is that we're going to have two homologous chromosomes. We're going to have two homologous chromosomes. That will be the first one. Then another one will be there. So we'll have two homologous chromosomes, which will be different, of course. And then each have two chromatids, which are these two guys at the bottom here. So those are the two chromatids. Each will have two chromatids. And then from there onwards, the two chromatids are gonna overlap, like the way they're doing here. They're overlapping to each other. Then from there onwards, the genetic material from each will be exchanged. Then the new ones will be formed will have a different mixture. For example, the new one that we're gonna form here will have it will be like this now. It will have the yellow part, this white part that we're having, and the dark part. So the remaining portion will be dark now. It will be coming back with the dark portion that it will get from the other one. Same thing as the other one. It will have the dark part and the light part from the other one. That's how it will happen during crossing over. So I'll repeat again. Initially, we're going to have two homologous chromosomes. These two like that. Each chromosome have two chromatids. And then the chromatids will cross over. This will lead to exchange of genetic material. Then from there onwards, the new ones will be formed will have a mixture of two different chromosomes. That will be the whole process of crossing over. So there are only 3 points. Even if you put 6, they'll mark the first 3, so you'll be fine. 1.5, we're going to genetics now. We're being told that we've got a black cat color, a cattle which is black coat, so that's B. So we've got a capital B as coat, black coat color. Is dominant, so that's dominant. So if it's dominant, meaning it must be capital B. Then yeah, that's shown to us there. We've got a brown coat, which is small b. Okay. We also have got long horns, also, which is big H, and short horns, which is small h. So we're having two different traits now. It's no longer a monohybrid; it becomes a dihybrid crossing. If we've got two different traits, it's what we call a dihybrid crossing. So state why the example above is dihybrid crossing, because we've got number one, two individuals who are being crossed. That's the first point. We've got two individuals being crossed. That's the first point. Two individuals are being crossed. So two individuals are being crossed. So we've got two individuals being crossed, and then they've got two traits. So they've got two traits, two individuals with two traits which are different. So they've got two different traits. So that will be why we call it dihybrid crossing, because we're crossing two individuals. We're having two different traits, and then here we know the traits will be coat, color, and also horns. So the two different traits, which is coat, color, and also horns in this example. So they've got two different traits, which is coat, color, and also horns. And then we know that they're controlled by different genes, with two different genes controlled by two different genes. So they're being controlled by two different genes, and our genes which are controlling them here is B and H. So they've been controlled by two different genes. So that will be your answer. We've got two different individuals being crossed, and these two individuals have got two different traits. Two different traits. Just put traits there. So they've got two different traits, and then those two traits are being controlled by two different genes, which is B and H. So the two different traits here is brown. It's coat, color, and also horns. Those are the two different traits. It's coat, color, and also horns. So we've got two individuals with two different traits, which is coat, color, and also horns. They are being controlled, and those two traits are being controlled by B and also H. That will be your answer for that one. That's why it's dihybrid. For mono, we're gonna cross individuals which are having only one trait. But if you're doing two different traits, that becomes dihybrid crossing. Give the genotype of a female parent. Okay, it's the female parent now. So the female is that one there. Or they're having a Punnett square. It's between a cow and a bull. Okay. Female are those ones who are up there. That's female there, and then male will be those ones who are down here based on the Punnett square. So they're asking us to determine the genotype of the female parent. Female parent, because we know we're gonna have, of course, there will be brown is dominant. There's gonna be brown dominant, so it will be brown dominant, but there'll be heterozygous, so it will be brown, which is dominant over small b. So the heterozygous, so it will be small b there, and then horns, they won't have horns because they'll have one big H and small h, so they're gonna be heterozygous for each. So it will be heterozygous for each. So they're gonna have brown coat and also horns. So that will be the female based on the different combinations I'm seeing here. So they must have each, but brown and horns are not dominant. Give the genotype of offspring X, or X will be these two. So for X will be these two. So for us to form X, the female will have BH, that's there. So it will, the female, just call it F there. The female will mate with the male. Male is coming with B and small h. These are the two that I'm going to focus on for us to form X. We'll focus on these two parents. These two parents. So we know for us to form parents, we're going to have that and that for X to be formed. So that will be those two going together. So you're going to have BB forming that one, and then from there onwards you're going to have H and H forming that one. So meaning it would be big H there and small h. So that will be the possible combination of X. So that will be our X for this question here. So that will be our X there based on that crossing. So X will be that. Then now let's go and find the possible one for Y. Y is this one here. So meaning for Y to be formed, it will be this one here. When this one and this one meet, they're forming that one. And then we're going to have that and that will form that. So now we're going to have that BH and forming that one. So we're going to have capital B and small h reacting with capital B and small h. Those two here, this one and that one there, so that we can form our Y. So from there onwards, we know again the same thing will happen. the genotype that will be formed will be big BB, which will be big B and big B, and also small h and small h. So the different genotypes are going to form. So that will be our genotypes. Oh, the phenotype, but the one that has to find the phenotype. Or the phenotype, remember, B is dominant, which is brown. A it's black, B is black, which is dominant. So meaning to be black, because big B is black, which is dominant, so it'll be black. And then remember, big H is horns, based on what they told us here. Big H is long horns. So meaning they've got small h, meaning they'll have short horns. So they will have short horns as the phenotype. They'll have short horns because they've got small h. They don't have a big H, short horns. So that will be the phenotype that we're going to form. It will be black and short horns. That's for letter B here. And then 1.5.4, what percentage of the offspring will be black with long horns? Okay, we need to go and figure out all the different possible combinations here. So we need to go and figure out all the possible combinations. So we need to do some bit of maths and calculating here, which is nice. That's why they always say that maths is everywhere, guys. Like, don't ever run away from maths. Maths will always be everywhere. So we need to find out possible offsprings who will be black and long horns. So meaning it must be those ones who will have one big B and one big H. So from, I'm just trying to erase this. I want to get the possible combinations you're going to get from the given table. So the given table, I don't know what I should figure out first. Now we can make a prediction. So remember, from this side will be big B and big B. So all the kids who are being formed here, so in other words, all the kids who are being formed in this row will have one big B and also one big H in them. So all the kids who have been formed will have 1, 2. You guys can go and fill it up if you want to be confident. All 4 kids who are being formed here are going to have one big H and also one big B, meaning all these kids who are forming here are going to have this genotype name. Just put here for you guys. All the kids who are forming in that row are going to have this type of genotype. We'll have big B and small b and big H and small h. So that all the kids will be formed in that one. So meaning we're going to have 4 kids. I'm going to have 4 kids will be like that in the first one. And remember, all these kids are going to be black with long horns. So because they've got the big B and big H. So all of them are black with long horns, those ones. Now let's go to the second one here. Second one here, remember, this will be reacting with all these 4 of them. So we know from this that all of them are going to come back with B, 2 BBs are going to come back. And then from there onwards, all of them are going to have small h and also small h. So all 4 that I'm forming here are black but short horns. So meaning they're not part of our study. So we'll ignore this 4 because we want only who will be black and long horns. These ones here are black and short horns. So they're not part of our question. So they're out. 4 is out. Now we're going to have this one here rating with this 4 here. Because remember, it will come back as one big B will be there and one small b will also be there. We're going to have one big H and one small h. And all 4 of them will be the same. Be one big B, one small a b, and then one big H, one small h. So all 4 of these kids are going to be also same what we're looking for. They'll be black with long horns. They'll be black with long horns. So all these 4 are black with long horns that will form here. Then lastly, big B and small b. So lastly, we're going to have this one's big B and small b there. And then they want to come back with small h and also small h. So these ones will be black, short horns. So they'll be black and short horns. So meaning all 4 of them are going to be black and short horns. And our question is only hearing that. In our question, we only want ones who will be black and long horns. So we've got 8 out of 16. So it will be 8 over 16 that we are having here, 8 over 16 possible kids are gonna be black with long horns. So meaning the aim is to get percentage. So to get percentage, you must multiply by 100. So meaning 8 over 16 times 100, that will be half. So it will come back as 50%. So we've got 50% chance of us having offspring who will be black with long horns. At the same time, we've got 50% chance also of having kids who will be black with short horns. They only ask us for black and long horns. That will be our answer for black and long horns. The answer will be the same also for black and short horns. But because they only want black and long horns, just leave it like that. But tomorrow, if they ask you for black and short horns, it will be those other four. The ones who have been formed here, those will be the black ones with short horns. These ones here. It will be 8 as well, over 16. So that will be our answer there. Then 1.5.5, which principle of Mendel applies to dihybrid cross only? Remember, that's the principle of independent assessment. It's the principle of independent assessment. Principle of assessment. So it's principle of independent assessment. So it's principle of independent assessment. Independent assessment. So the principles of Mendel, independent of assortment. It applies only on dihybrid assortment. That only applies in Mendel. So that will be that assessment. So that will be that for section A. So let's quickly go to section number B. So now we're going to meiosis. So I'm assuming, since I'm seeing homologous chromosomes, this should be meiosis 1. Based on me seeing homologous chromosomes, should be meiosis 1. So firstly, they're asking us to label the diagrams. So they're asking us what is X. X is this guy here. So remember, X is the cell membrane. So X is the cell membrane there. X is the cell membrane. X is the cell membrane. It's a cell membrane that every cell will have outside, especially all the animal cells, even plant cells will have that. That's cell membrane for X. Then they ask us for Z. Z are those things which are inside, those dots inside. We call them spindle fibers. We call them spindle fibers. We call them spindle fibers. We call them spindle fibers now. So we call those spindle fibers. We call those spindle fibers. So the dots which are inside the spindle fibers. And then 2.1.2, give 2 observable reasons why chromosome pair Y is regarded to as homologous chromosome pair Y. Oh, that's the one there. Why is it being called homologous chromosomes? Okay, remember, homologous must look the same, so meaning they must have same size, same shape. So they've got what we call same size, same shape, because they are homologous. So they must have same size. They must also have same shape, and anything that you can see the same size and same shape. Can be also length, can be any direction or color. You can say same color. Yeah, same color also will apply. So any two, it's fine. You can say they must have same color as well because they're homologous now. 2.1.3, identify the faces which are shown in diagram 1, 2, and then 3. So you want us to find diagram 1, this one here, diagram 2, and 3. So they want us to find all the 3 diagrams. So let's look at them now. One, yeah, they're in the equator. One, they're in the equator, and the one that is in the equator, that is metaphase 1. So remember, in metaphase 1, they must be in the equator. So during metaphase 1, they must be in the equator. So 1 will be metaphase 1 because they must be in the equator. That's metaphase 1. 2, okay, those ones, they look like they haven't exchanged. Remember, these ones have got different mixtures in them. So meaning they've underwent prophase already, these ones. These ones, they're like unique. They don't have mixtures of black and white in them. So meaning this is before metaphase. And before metaphase, we've got prophase 1. So that should be prophase 1 before they do crossing over. So that should be prophase 1, the first step, because metaphase is the second step here. So meaning must be prophase 1 because they are still in their original forms. They don't have any mixtures in them. Then lastly, we know they've got two different cells now. So we're no longer having one cell anymore. And if you've got two cells, and then with two cells should be telophase 1, but telophase 1 should be those homologous together. Here I've got different chromosomes and there are 4 of them. So whenever there are 4, it means it must be metaphase 2. Because metaphase 1, when I have homologous still together, can't be metaphase again. It should be so they're now no longer one cell. So now we're in meiosis number 2. And again they are in the equator, but now it's different chromosomes in the equator. So again they're in the equator, but different chromosomes. So that should be metaphase. But it can't be one now because now it's no longer homologous. So it's not now they're separate. And for them to be separate, it must be metaphase 2 because it's in the equator again. So the one that will be in the equator will be metaphase. But because the no homologous chromosomes will be metaphase 2. List 2 events in meiosis that will contribute to genetic variation. Okay, that will be crossing over as well as random arrangement of chromosomes. The 2 events that will cause genetic variation will be crossing over, which occurs in prophase 1. So that will be crossing over, which we know occurs in prophase 1. So that will be crossing over, and also random arrangement of chromosomes. So it's crossing over and also random arrangement of chromosomes because the chromosomes are arranged different. So it's also random arrangement of chromosomes. Those are the 2 ways that will cause genetic variation. So it will be random arrangement of chromosomes. Random arrangement of chromosomes. Okay, again we're going to another crossing. We've got a blue-eyed woman marries a heterozygous. It marries a man who is heterozygous for brown eyes. Okay, brown eyes are dominant over blue. So remember, if brown is dominant, well, as long as you see one big B, they become dominant. And then meaning blue eyes, for blue eyes to be formed, the mother must have small b, and there must be 2 of them. So meaning it will be that for the female. So the female will be small bb for them to be blue-eyed. Then the male is heterozygous. Remember, heterozygous must have 1 b, and also 1 small b. That will be the male. And then the female will be homozygous for the condition. That will be bb. Male who's heterozygous will be 1 big b and then 1 small b. These are the parents. Just call them P1 there, that they can see what you're saying. That will be the P1 genotype. There's a P1 genotype there. Just try to put them somewhere. P1 genotype. Just put it somewhere around there. P1 genotype. That at least it can be there. P1 genotypes. But at least that must be shown as well. Because when you guys are being marked, they're gonna look for such things. P1 genotypes. And then we know that phenotype. That's blue-eyed there. That's blue-eyed. That's blue, and then this one we know it's brown. That one we know it's brown. Those are the phenotypes of P1 or the parents. Those are the phenotypes. Types of the parents. Phenotypes. Those are the phenotypes. Those are the phenotypes. Okay, so we've got that as our phenotypes. Ooh, that came out wrong. So now we know that now our different genotypes are going to undergo what we call meiosis. So the different genotypes that we have from parents will undergo meiosis, which must be shown as well there. So meiosis will give us gametes. So we're going to form different gametes, which we'll have one as big B, the other will be small b from the father. Then we're going to have the mother having small b and small b. These are the gametes now, which have been formed by meiosis. So this will be the gametes formed by meiosis. So gametes which will be formed by meiosis. So this will be the gametes formed by meiosis. Okay. So now from there onwards, each will cross. We're gonna have that one and that one there. So remember, those 2 are gonna form big B and then small b. Then we're going to have that big one with that one. They'll form big B, small b, and then you're going to have small b and that one there. They'll form small b and also small b. Lastly, gonna have that one there and that one forming small b and also small b. So the whole process that's undergoing here, we call it fertilization. It's when the offspring are being formed. Just put everything there. That'll be fertilization there. That'll be fertilization there. That'll be fertilization there. Fertilization will be formed there. So from there onwards, we're going to form the F1 kids. So the F1 or the children. So the F1, firstly we're gonna put the genotypes there. We're gonna call the genotypes, the G, the genotypes. Just call genotypes. Yeah, genotypes. So the genotypes of the kids that will be formed. We're gonna have 4 different genotypes now. Genotypes of the F1 or the kids will be, we're going to have two big B, small b, and also 2 big B and big B. Those are the genotypes that are forming in the F1 kids. And then the phenotypes. Good to have the phenotypes of the F1 also. So there'll be 2 big B phenotypes there. So big B, remember, we're told that brown eyes are dominant. So meaning you're going to have 2 brown eyes. So you're going to have 2 brown eyes now. So you're going to have 2 brown eyes. So you're going to have 2 brown eyes. So you're going to have 2 brown eyes. And then also 2 blue eyes. And 2 blue eyes in the kids. So it's 50% chance of the kids having brown eyes, and also 50% chance of them having blue eyes. So that will be 2 blue eyes. So there's 50% chance that the kids will have brown eyes, and also 50% chance that they'll have their blue eyes. So those will be your genotypes and your phenotypes. So that will be your lovely answer for that one. Then now let's go to 2.3. We have been given a pedigree diagram which showing an inheritance of a genetic disorder caused by a single gene. Okay. And then 2.3.1, use individuals 2 and 5 to explain why this disease is caused by a recessive allele on the chromosome. So they want us to use 2 and 5. Those 2, that's 2 there, and 5. Okay, so we need to use those 2 to explain what could have led, or why this is an recessive allele on the X chromosome. Okay, number 2 and number 5. So let's check them. In the individuals 2 and 5 to explain why this is not that. Okay, so from this, square is unaffected, also female unaffected, male is affected, female is affected. So in other words, individual number 2 here has the condition. No, doesn't have the condition. Number 2, it's a female who's unaffected. Ne? So number 2 is a female who is unaffected. Number 5 is a male who is affected. Okay. So remember, for a male to get the condition, let's say the condition is something about which letter can I? Let's just use B for the condition. For the male to get the condition, if you've got small b, for example, and a y, they'll get the condition. And then the mother can be 1 big B and 1 small b. She won't have the condition, this mother. She'll be a carrier. This mother will be a carrier. So based on what's given to us, female number 2 must be, she's normal carrier. Ne? So female number 2 is normal carrier. Female number 2 is a normal carrier. She's a normal carrier of the condition. So female number 2 is a normal carrier of the condition. Normal carrier of the condition. That's female. So just so that this will be the possible genotype that should have that female in number 2. That will be the possible genotype that should have that female number 2. She's a carrier, this one. And because she's a carrier now, she had kids with a male who was normal as well. And then when they have those kids, they're having number 3 who's having a condition. Okay, they didn't say anything about that. That's fine. Our focus will be on number 5, who's one of the kids as well. So the 5 is a male who's having a condition. So first point will be that individual number 2 is a normal carrier, meaning they will have that genotype. So the recessive allele they have is on the X chromosome. So the recessive allele they have is on the X chromosome. So the recessive allele they have is on the X chromosome. So we must mention every point. Recessive allele number 2 has is on the X chromosome. X chromosome on X chromosome. Recessive allele is on the X chromosome. That should be another point you can put. So when she has kids with the normal male who's number 1. So when they have kids with the normal male who will be number 1, who's normal. So it will be a normal male and a female male. So at some point, the recessive condition that the mother is having, the recessive condition on the X. So meaning in other words, this XB that the mother is having, that small XB that she's having, number 2, she will pass that. She will pass that allele that she has to one of her kids. Pass to one of the kids, to one of the kids, to one of the kids. So she will pass that to one of the kids. So she will pass that small allele that she has to one of the kids. So one of the kids who's in this instant is number 5. We will have that XB because it's a boy. They'll have a Y chromosome. So whenever you've got a recessive condition on a boy, it will always show up. So whenever you have a recessive condition on a son, it will always show up because the Y chromosome will not be able to mask it. Only females can be carriers. Males will never be carriers. That's why number 5, this is number 5. That's why number 5 is having the condition because males can never be carrier of a trait that causes a condition. Number 5, he has got that recessive allele in them. That's why they're showing the disease. So first point, you will tell us that individual number 2 is a carrier or must be a carrier of the condition having that genotype then. She married a normal male who will have a normal genotype, which will be X, big B, for example, and a Y name. That's a normal male there. So one of their kids will have this type of genotype. And that X small b, which is a carrier or recessive allele, is showing in one of the sons who will be number 5. Number 5 is showing the condition. And the condition will show on number 5 because number 5 does not have 2 chromosomes. Only girls or females who are having 2 chromosomes can become carriers. Males can never be carriers. If a male has got a condition, it will always show up in them. There's no other chromosome which will help them. The Y chromosome will never help them. Females can become carriers because the other X chromosome will help them. But because male have got 1 chromosome, then they're going to get the condition. So meaning number 5 is getting the condition from his mother, who is number 2. That will be a conclusion. So again, I'll repeat. Normal number 2 is a normal female, but she must be a carrier with that genotype there. She married a normal male with that genotype. The recessive allele that the female is having is on her X chromosome. So the other chromosome that she's having will mask that recessive allele. But one of the kids that she's having will get the X chromosome with the recessive condition, this one. And then that condition, she's passed it to number 5. And number 5 is getting the condition because he's a boy. Because boys have got X and Y. So boys cannot be carriers. Those are the conditions showing on the boy. So number 5 will be X small b and also Y for the condition, because boys will never be carriers. So that will be answer for 2.3.1. Use the letters XN for the recessive allele and also X big N for dominant allele to give the genotypes of the individuals. Now we've been told to use letters N. Okay, that's fine. So because we've been told to use letters N now, let's just want to get some space to show the different combinations. So basically we've been told that small n is the one that causes the condition, but big N won't cause the condition. So small xn will be recessive condition there, then this one will be the dominant one. So we're being asked to determine the genotype for number 3. That's number 3. Number 3 has the condition. That female has got the condition. So it's a female with the condition. For a female to get the condition, remember, they must have 2 recessive alleles there. So it will be small n there also, and the X will also have small n because the female with the condition. Then number 4 is a male who's normal. A normal male will have X and a big N there because they're normal, and then Y, that will be the normal male. X big N and also Y, that will be the normal male. So remember, for the females to get the condition, the 2 recessive alleles must be present. For males, if they ever get one recessive allele, if you're a male, you get the condition. But if you're normal, therefore, the recessive allele must not be there. What percentage of daughters produced by individuals 3 and 4 will be affected? So 3 and 4 are having kids now. Okay, let's just do the crossing quickly. So we know 3 is having the condition. So it will be small n, and then other X also will be small n. That's number 3 now. Number 4, we know they are normal. So it will be X, big N, and Y. That's for normal. So the possible kids will have that. So it will be X small, okay, X big N and X small n. That's the first combination. Then from there onwards, you're gonna have that and that. It will be X small n and then Y, and then you're gonna have X small n. It's another possible combination. X big N and X small n, and then lastly we're gonna have X small n and Y, X small n and Y, the X small n and Y. Okay, so we're having that kid there and that kid. So the girls are normal. We've got 2 girls who will come out like this: 2X big N and also small n. Then also we're having boys who will have 2X and then small n and Y. So the 2 girls who are formed are normal. So in other words, it will be 50% normal, 50% normal and also 50% having the condition. So it's half-half. So the question was asking us what percentage of daughters produced by individuals 3 and 4 will be affected. All the girls which have been produced by 3 and 4 are normal. All of them are normal. So there will never be a girl who will have the condition. Only the boys will have the condition. So they're asking us what percentage of the girls will have the condition? 0. Because all the girls which have been formed are normal. You will never get a girl who will have the condition. All the girls who will be formed will be normal. Only the boys who will be formed will have the condition. So only boys will be affected. The girls won't be. So it will be 0% because the girls won't be affected. 2.4 now, we've got a group of grade 12 learners who are doing a survey to establish the number of learners with blood group. Okay, we've got high number with blood group A, small number B, and then AB, then O. So most of them are having blood group A. Okay, so number 2.4.1, state 2 planning steps that should be considered before carrying out the investigation. Like, remember, every time when you're doing an investigation, guys, you must always ask for permission. Like whatever you're doing, whether you're doing a survey in your classroom, at home, wherever, they must ask for permission. it's always good to ask for permission. You can just go and take people's blood without asking them for permission. So you must ask for permission. That should be a must. You must ask for permission. So ask them for permission. You can't use my blood without telling me that you want to use it. Next thing I see my results all over the news or social media platforms. So you must ask for permission, and of course they must have a venue to get the blood. So they must have a venue. Then they have a venue. They must have a venue, which will be a lovely place to do the investigation. So they must have a venue. My spelling of venue is so terrible. They must have a venue there. So, I'm writing with English FAL. So they must have a venue there. I'm doing too much English FAL. So what else can they have? Remember, you must at least have good record. Record must be kept. Like it must be accurate record. Whatever they're taking, it must be accurate record. So they won't ask for two. Like anything that must be feasible in investigation. Whether it's a physical one, physical science one, whether it's a math investigation, a Greek one, geography one, you must always ask for permission. And also, of course, have a venue. You must keep all the records. Yeah. Which blood group has the lowest percentage of learners? Okay, it's obvious from the graph. B is the one that is having lowest from the graph. That's B. So B is the one that is having the lowest. Give one reason why the data from this investigation was shown using the above graph. So remember, we are doing different blood groups. So whenever you're doing different blood groups, it's always good to use separate data. So one reason why the graph, we're doing different blood groups. So because we're doing different blood groups, it's always good to use it like that because they're different. You can't use the same type of like line graph, because line graph will make it difficult to spot which blood group is what. So mostly use this type of bar graph if you're using different stuff like different blood group, different age. So mostly use a bar graph for that. So the reason why I'm using a bar graph is because in the study we are studying different blood groups. So that's why we're using the bar graph, because we're investigating different blood groups. That should be your answer. What conclusion can be drawn from the results which are shown? So from the shown results which are shown, we've got blood group A. So we've got 45% of people are A. 5% are A. That's what we can see from the bar graphs. And then we've got 10%, that's B. 10%, that's B. And then we've got 20%, that's C. 20%, not C, why am I saying 20%? 20%, you, I don't know what I'm writing now. 20%, it's A, B, not C. I don't know where I got C from. So this thing of memorizing A, B, C. So 20% will be A, B. And then lastly, we're going to have 35%, that's O. Okay, 5% that's O. So that's what we're seeing from the results. So you can conclude that majority of people in the study were having blood group A. That should be our conclusion. So we've given evidence, and then we just conclude that no, from the given graph, majority of people were having blood group A, or the lowest, or fewer people are having blood group B. Yeah, but mostly I think it's safe to say that majority are having blood group A. And then 2.4.5, the frequency of each blood group worldwide is shown below. Okay, in the worldwide, I was about to ask why. Was mostly worldwide we've got more people with blood group O. So we call those ones with blood group O universal donors. The learners were expecting their results to correspond with the results in the table, but they do not. Explain why the results in the investigation are not reliable. Remember, the sample size was probably small because we've got 45. If we have got a problem with your results not being accurate, sample size small. So the reason that they could have gotten their different results was that they used a small sample size was small. Sample size was small. So never use a small sample size, you'll never get accurate results. So never use a small sample size small. example, if you're doing a study of only 10 people, your results will never be accurate because you're only using a small sample size now. So, and it's only for grade 12s. So maybe if they were to do from grade 8 to grade 12, maybe the results will be different. Because if they do grade 8 to grade 12, therefore they'll have more blood samples in them. But because they're only doing grade 12s alone, so it's a small population of grade 12s, that's why their results are going to be not accurate. So for them to have accurate results, they must include more people in the study. So they must include more people in the study. So your reason will be it's a small sample size. For them to have accurate results, they must increase the sample size, meaning they can do from grade 8 to grade 12. So that should be our lovely answer there. And then let's go to 2.5 now. We're going to evolution now. So we've been given different skulls. We've got African ape, Australopithecus, Homo erectus, and also Homo sapiens. The other one was 9 million years ago. That's a long time ago, guys. And then recent was around 0.2 million years ago. That's like 200,000 years ago, which is us. So recent. 2.5.1. How many million years ago did Homo erectus first appear? Homo erectus, there we are there. That's 2 million. Yeah, that's 2 million there. So let's start from 2 million. Should be 2 million, 2 meter house. All of us will have to have that in our bank account. 2 million in our bank account. 2.5.2. Calculate the difference in brain volume between Australopithecus and Homo sapiens. Okay, in brain volume. So we're focusing on these guys here and Homo sapiens, these ones here. So I want us to find the difference in the volume. Oh, the volumes are here. The brain volume is this one. Okay, Australopithecus, we know it's 430. So the difference, remember difference in maths, it's minus there. Difference will be minus. It's 430. And you can't do 430, but that will be negative. So start with the bigger one. The bigger one is for 1.4. You can't do a negative number with such brain size. Imagine having a negative brain size. So it'll be 1.450 minus 430. So that'll be the difference. 430. So let's do that. 1,450 minus 430. 1,450 minus 430. Okay. That's a huge difference. 1,020 centimeter. So that's a huge brain difference. So meaning, so the difference is huge. So meaning our brain is huge because that's our brain size now compared to these guys. That's a small brain size. And also, African ape was having a bigger brain size than Australopithecus. What happened to you guys, Australopithecus? Why were your brains becoming small? What was happening during that time? When the environment making them having smaller brains, or they're not thinking well, because you can't have African ape having a bigger brain than these ones. They have a smaller brain. Maybe they're becoming lazy. These ones. Then yeah, Homo erectus came in and the brain size doubled. Then we have us now. We've more than tripled from what Australopithecus was having. So that's the difference in brain size. That should be your answer. State 2 advantages of having a large brain. Okay, having a large brain, of course, guys, you can think fast. We can think fast. We can do things like thinking fast. We can think fast because as we're thinking now, we are doing things like AIs. We are multitasking. We are doing a lot of things, guys. Having a smart brain is good. You can do 7 subjects in one day in the classroom because of having a bigger brain. We can also it also allows us, besides think fast, we can also speak a lot of languages. Remember, I'm Fenda, someone is Zulu, someone is Tonga, someone is Ndebele, someone is Xhosa, someone is colored, someone is from white speaking nation. So we can speak all languages, like a lot of languages, so that our brain can store a lot of languages. Because an average South African should at least speak 5 languages. I can speak 6 fluently, so because the brain is good for us to to be able to speak a lot of languages. So if you are normal South African and you can't speak more than 5 languages, I think personally you should be a bit not ashamed of yourself, but you really should challenge yourself a bit. You can be proud of only speaking 3 languages or 2. No, guys, you need to improve. We've got a large volume of brain. You need to start speaking a lot of languages now. Our brain is huge. 2.5.4 tabulate three visible differences. Okay, tabulate. Oof, I need space for this. So into tabulate. Remember, there's a mark. Remember, whenever you hear the word tabulate, guys, I always advise learners. Whenever you hear the word tabulate, you must know there'll be a mark for a table itself. Because most learners tend to write answers in a point form or whatever. And that is wrong, because once you do answers in a point form, you end up losing free mark, which is for the table. So whenever you hear the word tabulate, firstly, put the table. Before you even do anything, when you hear the word tabulate, just put the table, and you must use a ruler. Don't be like me. Use a Knafler ruler, and then put a table. Then you get a mark for the table. But of course, the table must show Homo sapiens, and also they're asking us for Homo sapiens and African ape. So that's Homo sapiens. That's H sapiens. So of course, your table must have that H sapiens. You must show that in your table. You can just go and put a table and leave it as blank. Then I don't feel like you guys don't deserve a mark if you just go and put a table like that. And then we've got African ape. African ape. I wonder how these guys were like back in the day, eating raw meat. Evolution is nice. Okay, Homo sapiens. We need three differences now. Okay, we need three differences. We need three differences. So Homo sapiens, that's us here. So you can see we're comparing you and that one. So we're comparing the two of us. Remember, us, we've got a bigger brain. So for us, it will be a large cranium. Remember, the skull of the brain is cranium. So we've got a large cranium. Our cranium is bigger. So we've got a large cranium. That should be our first point. We've got a large cranium because our brains are bigger. So meaning that one will be small cranium. The other one will be small cranium. That's the first difference. The other one will be small cranium. The other one will be small cranium there. Because that's the difference, first one between us, that we can see from the diagrams also. And also we can see African ape, they've got a big jaw. So that big jaw, so they've got a big jaw. So just call it a big jaw there. They've got a big jaw there. So us is a bit smaller than them. Some do have a big jaw, but generally most of us have a smaller one. Some do have a big one, because I always get people arguing, no, I've got a friend who has a big jaw. Yeah, but can't be as big as the small ape, as the African ape. Us are mostly smaller. And then also remember, they were eating raw meat. This one, look at the teeth. They were eating raw meat. So meaning they had large canines. Canines were there to eat raw meat. So they're large canines that was there for eating raw meat. As we are eating well-processed food with spices, food which is soft. So we don't need large canines. So we've got small ones. So you just need three differences, guys. And such questions, I feel like every learner in the whole classroom must get, guys. You can't afford to lose such precious marks. Having a mark for the table and only three differences, which you can all see in the skulls. You can all see these differences between the two. This is the bigger cranium there. This is small. The jaw is big here. The jaw is small. The teeth are big here. The teeth are small, or the canines. That's it. And then describe two structural features that correspond with evolution to bipedalism. Remember, it's us who are working with 2 feet now. So it's us who are working with 2 feet. Remember before we had that magnum that you guys must get after writing your November exams? So we've got the foramen magnum. So the foramen magnum again is back. That is that hole that you have there, which can go forward and also backwards. Foramen magnum. So for bipedalism, it will move backwards. Foramen magnum. I'm spelling it so weird. Foramen magnum. So it will move backwards. Foramen magnum will move backwards. Foramen magnum move backwards. That should be the first thing that will allow us to move bipedalism backwards. So it's by we're moving with 2. So it will go backwards now. Foramen magnum will move. No, it will move forward. Move forward, not backwards. Backwards is for those ones who are using four limbs. So foramen magnum will move forward. It will move forward. Because for us to move with 2 legs, it must move forward. For us to move with two legs, it must move forward. It will move forward. That's the first one, foramen magnum. And then also remember, we also have got our pelvis, that hip bone. So the pelvis also will go from narrow and long to become wider and short. So the pelvis, it will go from long and narrow. So we'll go from pelvis will go from narrow and long, go from long and narrow, from long and narrow. Now it's becoming more flexible for us to move now. Long and narrow. So we go from long and narrow to long and narrow to, it won't be long anymore, so it becomes shorter. So it become short and wide. So it becomes short and wide. So those are the 2 points which will help you guys. Short and wide. So it'll be short and wide. So those are the 2 points that will cause us to become bi wide pedanism. So those are the 2 points. And also the spine will go from C-shaped to become so it will go from C-shaped to become S-shaped. So the spine will go from C-shaped to become S-shaped. That will be the third point. The spine will go from being C-shaped to become an S-shaped. So those will be the 3 features that we're going to have. So the spine will also go from, the third one will be, number 3 will be spine will go from C-shaped to become S-shaped. That will be the third point. 2.6, now we're going to GMOs, genetically modified foods. So they're bringing, I don't like these foods, guys. I feel like they're making us so fat. Guys, I feel like all of us are becoming so huge because we're eating a lot of junk because of these foods. I know they help with food shortage and all that, but they're becoming so fat. So there are also many objections. I'm one of those people who are objecting them. They're lovely, they taste so nice. It's nice eating a burger and all that, but guys, no. We're eating poison. No, not poisons, but I feel like in the long run, we'll regret eating this food. Give one reason why the initial cost of production of GMOs is high. So remember, guys, we're modifying food. So if you're modifying food, therefore, whatever you're using to modify the equipment they're using, the equipment should be expensive because not everyone can afford to modify them. So whatever the equipment they're using to modify food is going to be expensive, because if they make it easy for everyone to modify, then the whole world will full of food which are modified. So equipment is expensive. So equipment is very expensive. That's why it's tough for many people to just make GMOs any day. So the food will be very, very, very expensive, the equipment. And then also remember, for food to be approved, it must undergo trials. And I think most recent there was a trial where they were trying the COVID vaccine. It took so long because trials take too long, guys. Before something can be approved, it must undergo a lot of testing. So trials take too long. So that's why it's tough for GMOs to have to be done easily, because they must undergo a lot of trials, and trials take too long. That could be another reason that you can use. GMOs are considered a threat to biodiversity. So why are they being seen as a threat? So remember, they are going to a normal environment. If I'm having an animal which is GMO'd, it's going to end up meeting a normal one. And the GMO, there's a big chance that a GMO'd animal will be strong. If I'm going to make, let's say, an impala, a GMO'd impala, there's a big chance that it will run faster than the normal one. If my GMO impala is faster than a normal impala, therefore it will always outrun my normal ones. So in other words, these GMOs ones will always outcompete the normal animals, for example. So they will always outcompete normal ones. So they're always going to outcompete, because geo mode ones are made better. So they're gonna come there with a bigger speed. If it's a lion, it will come there with bigger teeth or bigger jaws. So they'll outcompete normal animals. So that's why they're a problem. Because now if they're outcompeting normal ones, therefore all normal ones will end up dying now. Because normal ones will have like the big advantages that these ones are having. So that should be your answer. So it's only 2 questions for that. Okay. Now let's go to question number 3. So given a long paragraph, we've been told that scientists have studied the appearance, genetics, and behaviors of wolves and dogs. Oh, they're comparing wolves and dogs that we're having at home to better understand what caused them to diverge. Okay, so they want to know what led to wolves becoming different from wolf sporty that we're having at home. You know, wolf sporty, woman to, yeah, different names of dogs we have at home. So wolves began to evolve through a process in which people selectively bred individuals with desired, okay? So this will lead to reduced aggression and willing to submit to human social structures. Scientists have discovered that some wolves have had genes to produce amylase, okay, and digest starch, and then this benefited from the human food they were scavenging, okay. Domestic dogs produce much more amylase. Oh, okay. So the dogs have got more amylase in them. That's why I was spotted they can eat more of our food than wolves. So wolves, they cannot eat more of our food. Okay, that's a good story. So this allows dogs that we have at home to eat everything. If you leave your Simbas, they can eat them because they've got amylase to break whatever we're eating. That's why our dogs can eat everything. But wolves tend to struggle. I've always wondered as well, if I go to wolf and give it the normal food like your quarters and all that, because some dogs at home they can eat cod. I wonder if a normal wolf can eat that. I don't think so, but it's something that I'm worth trying to see if a normal wolf can eat that. So 3.1.1, using example given in the passage, describe how a population of wolves may have evolved to form first population of dogs. So they're asking us how the wolves could have evolved. So they're basically asking us to explain natural selection. So of course, the first point, there must be variation in wolves. One of the wolves would have been different from the other ones. There should have been a variation in wolves. Should be the first point. There should be variation in wolves. So there should be variation in wolves. So there should be variation in wolves for them to start developing or changing. So there should have been variation in wolves. Some of the wolves would have been different from the normal dogs. So there should be variation in wolves. And that will be the first point. So this could have led to, based on the paragraph, reduced aggression. This could have led to reduced aggression based on the paragraph, or it could have been reduced eating of food, or whenever the changes could have been occurring. So it could have been reduced aggression based on their behavior. So you can use behavior or eating of the food. And then this also, remember, also we know ability to amylase. So they had a lesser chance of producing what we call amylase to digest starch. So they had a less chance of producing amylase. So amylase will be less in them compared to a normal dog. So amylase will be less in them compared to a normal dog. So therefore, this population will be having reduced aggression and amylase, then population it will separate now. So this population will start going different ways now because we can't produce amylase. We've got reduced aggression, then they start go forming their own group now. Maybe they think that other ones are special, so they start forming their own group because they think that the other ones with more aggression are not the same as them. So the population will start separating based on that. Population will start separating, and then once they start separating, remember they're not forming amylase, so they become scavengers based on the paragraph. So they become scavengers because scavengers based on what they're telling us in the paragraph. So they become scavengers. So because they cannot eat what we're giving them as humans, they become scavengers because they can't produce that amylase. They become scavengers. So meaning they can't live with us humans anymore. They can't live with us humans anymore. Can't live with us humans. Can't live with Homo sapiens. So they started moving. They can't live with us anymore. Can't live with Homo sapiens or humans. They can't live with us humans anymore. Based on that, so they can't live with us humans anymore. So they start separating. They can't live with us anymore, and then they start having genes to act for scavenging life. So they start developing more genes for scavenging, genes for scavenging. So they're gonna have genes for scavenging. So they'll have genes for scavenging. So they're gonna have genes for scavenging. So they'll have genes for scavenging because they cannot live like human being. And then the F1 offspring will be born with that trait. So all the new F1 offspring which have been born, they're gonna be born with the new trait of reduced aggression and also scavenging. So the new F1 offspring. That's why the wolves became a new population. So the new ones, this F1, they will be able to adapt to the environment of scavenging. The F1 will be able to adapt to the environment of scavenging. So that's how you're gonna do them, but in a point form. So you need to mention that there was variation in wolves and the normal dogs, and then the wolves were having a reduced aggression and also less ability to produce amylase. And then now, based on that, the population starts separating, and then now wolves, because they can't form amylase, they start scavenging, and then now they can't live with us human beings anymore because they are relying on scavenging. And then now the kids who are being born will be born with genes to survive the environment of scavenging. So now that's how all the new kids which have been born will be having the ability to scavenge. So I think they'll rank you for any 6 points that we'll be giving them. 3.1.2, although all dogs are descendants to the wolf, the use of artificial selection has allowed humans to drastically alter the appearance for dogs. For centuries, dogs have been bred for various desired leading to range of dogs from tiny Chihuahua and the massive Great Dane. So they're breeding dogs to form different forms. Like here, you've got a smaller one, you've got your bigger ones like your police dogs and whatever. So explain why all breeds of domestic dogs belong to the same species. So they're asking us to explain why all breeds will belong to the same species. So remember, for them to be species, remember how we define species. For them to be species, they must be able to interbreed. For them to be species, they must be able to interbreed. If you bring any type of dog, they will always interbreed. Doesn't matter whether it's a police dog or Chihuahua, dogs will always interbreed. For those ones who've got different dogs at home, you can see that during mating season, all dogs will be able to interbreed with each other. Like all dogs will be able to interbreed with each other, whether it's bigger one or smaller one, they will always interbreed with each other. Like that's how dogs are. So for them to be the same species, they must be able to interbreed with each other and also produce a viable offspring or fertile offspring. So they must be able to produce a fertile offspring. So that's how we define species. So if people belong to the same species, those dogs must be able to interbreed with each other and produce a fertile offspring. And all dogs are able to interbreed with each other and also produce a fertile offspring. So if dogs can do that, we can conclude that they belong to the same species, because that's how we define species. They must be able to interbreed together to form a fertile offspring. So that's how we can conclude that kid or offspring. So if they can interbreed with each other, same as human beings, whether you are huge, whether you are short, if you guys can interbreed and produce a fertile offspring, you are same species, same as dogs. That's why we can conclude that all domestic dogs are same species, because they can interbreed with each other to produce a fertile offspring. Letter B, describe how artificial selection has led to different breeds of domestic dogs. So remember, during artificial selection, humans choose what they want. So humans will choose the desired characteristics. So during artificial selection, humans will choose the desired characteristics. and then meaning they can breed the dogs of any phenotype that they want. So if I want to produce police dogs, I can only breed police dogs together. Therefore, I will only form police dogs. If I want to form only chihuahuas, I can only breed them as a human being. Then I will only form chihuahuas. So during artificial selection, you choose what you want. So your answer will be human beings will be able to choose the um, required or values or characteristics that they're looking for. So human beings will be able to choose the required characteristics. That'll be the first point. And then meaning they can decide the phenotype and the genotypes they want to produce. Meaning you can form, if I want to form only police dogs, I'll form only police dogs. If I want to form sporties alone, I'm going to form only sporties. So during this selection, human beings will be able to choose what they want. 3.2 now, the diagram represents the changes in a population of bacteria as a result of exposure to an antibiotic over time. Okay, we've got bacteria being exposed to antibiotic. We've got low, and then we've got that, and then that, which is dark. So we've got two different type of resistance there. We've got ones which are like light, and then ones which are having those lines, then the darker ones. So in diagram A, let's just count how many we're having, which are normal, like without any shading. We've got 1, 2, 3, 4, 5. We've got 5 which are normal shading now in diagram 1, and then we've got 3 which are like that. That's 3 of them are like that, and then we've got 3 also which are shaded. That's in diagram number 1. In diagram number 2 now, we have more shaded. We've got 1, 2, 3, 4, 5, 6. Okay, let me count again. 1, 2, 3, 4, 5, 6, 7, 8. 8 which are shaded in diagram number 2. 8 are that number 2, and we've got 4 which are like having like that. So no longer have no ones which are not having anything. So we know the first one is low antibiotic, then medium, and then high. So in diagram number 1, we had more which were having low antibiotics. That's in number 1. We had more which were having low antibiotics. In diagram number B, so in diagram B, we've got more which are having high antibiotic resistance. So meaning, in other words, before I even look at the question, in the first diagram, we had more bacteria which were having low resistance. And then at some point, the population of bacteria started having more, which are having high antibiotic resistance, meaning the antibiotic that we're using in diagram number A will no longer work in diagram number B, because most of them in B can fight the antibiotic. So that's what they're asking us. Explain in terms of natural selection the changes which occurred to number B. So now, remember, in the previous one of dogs, we had variation. So the variation there was due to amylase. Here the variation can be due to mutation. We can have variation due to mutation. Due to mutation, we can have variation. So due to mutation, we'll have variation. So due to mutation, we're going to have variation. So due to mutation, we'll have variation. So variation will come due to mutation. I put in a bracket variation, which is due to mutation. So we have that variation due to mutation, and then from there onwards, this mutation will lead to bacteria in B having antibiotic resistance. So it will lead to bacteria in B, bacteria in B having resistance, bacteria in B becoming resistant. So this mutation will lead to bacteria in B becoming resistant. In B, it will become resistant. That's in B, resistant to antibiotics, and then others not. So we're gonna have some in B being resistant to antibiotics, others not. Others won't be. Others won't be resistant. Others not. Others won't be resistant to it. And then this will lead to variation. We can also include variation again, variation in bacteria. So this will lead to variation in bacteria. So we're going to have variation in bacteria. So we're going to have variation in bacteria. This will lead to variation in bacteria because you're going to have one who are resistant to antibiotic and other ones who won't be. So this will lead to a variation in bacteria. So when antibiotic is used now, so I mean when I use antibiotic, when we use the antibiotic now, antibiotic, so whenever you use antibiotic, it will kill some bacteria and not kill other ones. So those ones who are resistant won't be killed, but the ones who are not resistant will be killed. Antibiotic will kill others now. It will kill others and not kill other ones. So antibiotic will kill others and not others now. So it won't be able to kill all of them. It will be able to kill other bacteria and then other ones will survive now. And then others will survive. So the ones who survive will be able to breed and form new F1. Survivors will breed and form new offspring. Breed and form new offspring. Breed to form F1. So they will breed and then they will form F1. And then all the F1 which have been formed now will adapt to the antibiotic. So all the F1 which have been formed will adapt to the antibiotic and then gonna form a lot of kids. Then this will lead to a lot of bacteria being formed, which will be resistant to the antibiotic. So firstly, we had variation, which will be caused by mutation. So we have got bacteria population, but some will be different due to mutation. Then when you go to diagram number B, we're having bacteria which are showing that they've got high antibiotic resistance, and then some don't have high antibiotic resistance. So now, due to the ones who are having a large antibiotic resistance, therefore, whenever the antibiotic is being used to them, it won't kill them. So they're going to survive. So the ones who are surviving, they're going to breed and produce an F1 and make a lot of kids. So all the kids who are being born now will be antibiotic too, so will be resistant to the antibiotic. And the more we are forming kids who are resistant to the antibiotic, at some point the whole population will end up being resistant to antibiotic, meaning now for us to kill the bacteria, we need to go and get a new antibiotic because the one that we're using currently is no longer relevant because all the bacteria will be resistant to it. 3.2.2 explain why antibiotic resistance can become a severe strain to the hospital. Remember, you can't use the one that we're using if all bacteria are resistant to it. You need to form a new one. So meaning they need to do a lot of new trials and a lot of money must be used because the one that is used now is no longer relevant. So they need to go and develop new ones, which will cost a lot of money. That should be a first point. They must go and develop new antibiotics, which will cost a lot of money. Also, some people will still be in denial. For example, someone will be like, no, I make this antibiotic, it will always work. Meaning they'll be forced telling patients to take large dose. So meaning we'll end up giving an overuse of antibiotic because the aim will be to kill the bacteria. But then if they give you, let's say, 1 spoon of antibiotic and then the bacteria doesn't die, now tomorrow they will tell you that no, instead of using 1 spoon, use 2 spoons. So meaning we're overusing the antibiotic. And then other point will be, remember, the more you are having resistance occurring. So let's say today we make antibiotic A. The next week, antibiotic A is no longer relevant. Next week, we're making antibiotic B. So now we're making a lot of antibiotics in a very short time. So meaning the government will be spending a lot of money and people are coming in large numbers because the more people become exposed to bacteria, the more everyone becomes sick. So those will be your 3 points. And first point will be making of antibiotics is expensive. It will lead to a lot of expenditure being used. Also, this will lead to an overuse of antibiotics. If you've got 1 spoon that was being used before, now they'll be telling you use 3 spoons, meaning people are going to be overdosed with antibiotics. Thirdly, we need to create a lot of different antibiotics all the time, meaning it will be strained for the country or the government to keep on supporting such trials. 3.2, read the following extract. Okay. Since 1972, biologists Peter and Rosemary Grant studied the finch population in the islands. They studied one population on an island called Daphne Meyer. The table shows the data they found over the last 7 years. Okay, so it was over the period of 7 years from 1974 to 1980. That's a long time ago, guys. More than 40 years ago. Using the information in the table, draw the line graph. Okay, don't have to use the table to draw the line graph. So for us to draw the line graph, first of all, have the different axes. So this will be time. You're gonna have time here, which will be your x. So you need to have time as your x. So you need to have all the different axes. That will be time as your x. And then on your y, we're investigating, or the us to find line graph show the number of finches. So y will be number of finches now, because of good rainfall. Y could have been rainfall, could have been small seeds, but they want us to only focus on number of seeds. So meaning our y will be number of seeds now. So there'll be number of seeds as our y-axis there. Number of seeds as our y-axis. Your handwriting so terrible. Then from there onwards, you only focus on this section for number of seeds. So number of years, we know it's from 1974, and then just want to show you guys how it will probably look like 1974, and then you're gonna put 75 until 1980. I'll be able to squeeze everything together here. So you guys will be given a graph paper, so it'll be 1980 there. Then from there onwards, same as you need to get a scale for number of finches. Our highest value for number of finches is what? It's 1,300. Okay, lowest is 250. So I think you can start from 200. Yeah, you can just scale of 200 as the lowest value you can use. Because the highest is 1.3 now. So we can have the highest as 1.4 there. And then just need to go and put everything as it is, because I won't be able to squeeze everything together here. Then from there onwards, you just need to plot them. The first value will be for 1980 is 250, which will be somewhere around there as my dot next to 200 now. And then 1.3 is in 1975, which will be somewhere around there. 75 will be around here. So it will be 75, which will be around there. Then you're going to put other ones values as well. And then your graph will be a line graph. So meaning you just be probably going up and down. Let me also put for 1979. 79 is 300, which should be around there if I'm using a good scale. And then 78 will be 350, which will be somewhere around there. And then in 1977 was 200. Oh no, it's going down now. It should be somewhere around there. 77, it's going down. That's 200. And then 76 was 1,100. 76 was up there. So it'll be somewhere around there. And then 75 was 1,300, which is there. So my graph will look like this. If I'm using an accurate scale, it will be going up, then it will go down, and then it will just remain a bit stable. That will be my shape. And then of course I must have the heading, which is number of finches. Number of finches, number of finches. That will be my shape. The number of finches, number of finches over time now. Yeah, number of finches. You can say from 1974, from 74 until 1980. Yeah, till 1980. That's fine. Okay. So let's go to 3.3.2 now. In which year were the largest drop in rainfall, number of seeds, and number of finches recorded? So we need to go and look at 1 year where we're going to focus on those 3 things. So we need to check the largest number of drop in rainfall, finches, and also seeds there. So we need to go and check which year was having the largest number of drop, or the lowest number. Yeah, the lowest number. Which year was having the lowest number? 1974, there's only number of finches there. 75, there's nothing about rainfall, so we can't use those ones because there's nothing. 76, we've got 130 small. Okay, finches were huge. Okay, also huge. 77, we had 20. Okay, that's a low number. It's a low number of rainfall, also low number of finches, and also a low number of small seeds. So the way that is showing largest drop. Because remember, from 76 we had 130. Then from rainfall we had 130. Then went to 20. So therefore, rainfall dropped from 130 to 120, which is 110. Number of finches dropped from 1,100 to 200. Which is a huge drop. Small seeds dropped from 600 to 90. So yeah, 77 was the largest drop that we had. Because other ones were not big drops. Because 25 was 1,300 to 1,100. That's a small drop. But from 1,100 to 200, from 600 to 90, that's a huge gap. So the largest drop that we got was 1977. That's when we had a huge drop in all those three things now. 1977. Explain how the events mentioned above occurred. So first of all, rainfall. So remember, for us to have a drop in rainfall, it must be drought. Currently now we are not in a rainy season. So whenever you get drought, that will lead to drop in rainfall. So there's drought. So for us to get a drop in rainfall, that will be drought. So drought will lead to a large number of rainfall. That's drought. So for rainfall will be drought. So for us to get a drop will be drought. And then the drop in number of finches. Number of finches will be food. So finches will be drop off in finches will be less food is available for them. So for finches to die could be less food, or the weather became unfavorable. Could be food. So we've got less food for them to eat. So less food. So for them to eat, less food, and also bad environment for them. So this could have been due to bad environment for them, or the weather, or bad climate. It's called bad climate. So that could have led to finches dying or decreasing. Bad climate. That could be the reason for those two. And then lastly, drop in small seeds. Drop in small seeds could have been decrease in plant growth. Decrease in plant growth. This could have been due to decrease in plant growth. Decrease in plant growth. Decrease in plant growth. Decrease in plant growth. So those three factors could have led to those three decrease in numbers. So decrease in plant growth. So those three factors could have led to that. So those three could have led to those three factors. 3.3.4. When the large number of finches decreased, there were still plenty of large seeds on the island. What does this tell you about the seed eating a bit of the ones who died? So meaning when the large finches died, we still had plenty of large seeds. So meaning the ones who died could not eat large seeds. Yeah, so that should be the reason. The finches who died could not eat large seeds, meaning they are pigs could not eat large seeds. So the ones who died could not eat large seeds. That's why I could explain why we still had large seeds in the island. So the finches were dying, meaning that they couldn't eat the large seeds. Maybe they only wanted small seeds available. So them not eating large seeds could have led them to die in there. So the reason would be they were dying because they couldn't eat large seeds. Maybe the pigs that they were having couldn't that it could not penetrate the large seeds. Then let's go to 3.4. Now we're going to photosynthesis. We're going back to DNA coding and life. So we're given stage W there, then there's molecule Y there, then there's stage X. There's also organelle 5 there. We've got different Z there, we've got different base pairings and all that. Okay. So firstly, they're asking us what is molecule Y? Molecule Y is this molecule that's formed here. So remember that's inside the nucleus. So in the nucleus, that's when you're going to end up forming mRNA during transcription. So mRNA will be formed during transcription. So Y should be an mRNA because the other one will be DNA. So this one that we're having here will be our DNA. Then Y will be our mRNA because that's in the nucleus. So meaning the HW will be transcription. So molecule Y should be mRNA. Then now they're asking us what is organelle V. Remember that during translation, and the organ that is there during translation is what we call ribosomes there, which is there to help in the formation of proteins. So that would be ribosomes, which help in the formation of proteins. So that organelle there should be ribosomes, which will help in the formation of proteins during translation. Then lastly, what is structure Z? So okay, remember this is transcription. So the mRNA that's formed during transcription will then move out of the nucleus through the nuclear pore. Yeah, that'll be the nuclear pores there. So meaning if that's the nuclear pore, Z should be the nucleus. Look at the nucleus. The nucleus is the whole thing. Should be the nuclear membrane. Yeah, it should be the nuclear membrane. So that'll be the nuclear membrane. Then the hole that is gonna go out from the mRNA, that will be the nuclear pore. So Z should be nuclear membrane. That's where the mRNA will live after transcription to go to cytoplasm to form proteins during translation. 3.4.2 give the sequence of nitrogenous bases for the first codon of molecule Y. First codon of what am I doing? First codon of molecule Y. Okay. So molecule Y we know it's TCA from DNA. That's TCA from DNA. So this one now, the TCA from DNA, we know when it forms mRNA, T will be with A in mRNA. That's C, ne? So C will be with guanine. Then A, remember, just be careful, in mRNA, there's no thymine. So meaning A will be with uracil now. So it will be AGU. So the answer for the first form Y will be AGU. If it was a DNA, it would have been AGT, but because it's an RNA, it will be AGU because you can't form T in so it'll be AGU as our answer. And then okay, 3.4.3 give the correct order in which molecules P, Q, R, and S will attach to molecule Y from left to right. So that has to form the correct order from P, Q, R, S. These guys were coming here. P, Q, R, S. That has to put in the order in which they will attach on molecule Y. Ne? So let's do them. One. P is U, UA. That's P. Ne? So for P to attach to U, our U, not our U, man, our Y must be, it's A, U will go with A, ne? So it'll be A, U. So it must be AAU. So the one that will form AAU, remember, here we know the first portion here, we already know it's gonna form, just put them as they are there. I'm putting the mRNAs which are gonna form them for each. For T, okay, I think I'll just put them here. Let's have TCA because I want to find the order in which those ones are gonna go. TCA will form this mRNA, and then let's call this one mRNA here. And then here I'm gonna put tRNA in it. So here I'm gonna put tRNA. And then another one will be C, J, T, and then we're gonna have T, T, C, J, T, TTA, and then lastly we're gonna have C, T, and A. That's our DNA one. So the mRNA that will form from this will be T, we know it will be with A here in mRNA, and then C will be with guanine, that will be J, that's guanine, and then A we know will be with uracil in mRNA, and then C will be with guanine, and then guanine will be with C, then because it's T there it will be with A in mRNA, and then because it's T there it will be with A in mRNA, then it also be with A in mRNA, and then A will be with U, and then C will be with guanine, T we know it's going to be with A will be with U in mRNA. Then from there onwards, the tRNA that will form, this will be the tRNA. The tRNA that will form here, A will be with U, and then this will be C, and then this will be A, then this will be C, J there, and then this will be U, CJU, and then this one will be U, U, and then A, and then this will be C, U, and then A. So this will be the PQRS that are coming. So let's find out how they are in the order. First one that will be there will be UCA. UCA is R. So the first one that will come will be R there. Then the other one will be CJU, which will be S. This will be S. The other one will be UUA. UUA will be P. Will be P there. And then lastly, we're gonna have CUA, which will be Q. So that will be the order they're asking us. This one here. So the order they're gonna attach will be R, S, P, Q. So my advice is do the DNA first, which is given to us already. Then do the mRNA that will form from the DNA. Then do the tRNA that will form from the DNA. These guys, PQRS, are your tRNAs. So you need to find out how the tRNAs are binding to the mRNAs. Then it will become easy for you to figure out who's going to be what and what. Where in a cell does stage X, okay, where's stage X? That's you there. Remember, what stage W is in the nucleus. Stage X will be in the cytoplasm. That will be in the cytoplasm. That's X will be in cytoplasm. That's during translation. It's in the cytoplasm. Be cytoplasm there. That will be cytoplasm. And then 3.4.5, if the natural basis A of the DNA strand was replaced with J, okay, describe how this will affect the protein that will be formed. If the third nitrogenous base of DNA strand, or the third one of the DNA, so before, okay, for this one, before we had T, that's the normal DNA, it's called DNA there. So the normal DNA that we have normally, this should be your answer. You can just do them like this now. Normal DNA, we have TCA as our normal DNA. We have TCA, that's our normal DNA as TCA. And then now the mRNA, or the new one, which is undergoing a change. So the new one now will be, they're saying that we're going to have a change of A. So A will be replaced now. So it will be TCJ. So the new one that's been formed now, it will be TCJ based on this replacement that underwent or occurred. So before we had the normal DNA of TCA, now we're forming TCJ. So the mRNA, let's just show how the mRNA would be also affected. So we're gonna have, this will affect our mRNA. So mRNA, the normal mRNA that we formed before of TCA, the normal one was AGU. Our normal mRNA was AGU, but the new mRNA that we're gonna form now will be A. Can I put A correct? So the new mRNA that we're gonna form now will be A. The new one will be A, and then J, C. So now, instead of forming AGU, we are forming mRNA of what? AGC. You must explain those things as point form. The normal DNA we had was TCA, but due to that change, we're forming a new DNA of what? TCJ. The normal mRNA which would have formed would have been AGU, but because of that change, we're forming AGC. So now the AGC that we're forming, that AGC that I'm forming now, let's find what its tRNA will look like. So that's our mRNA. So the tRNA we're gonna form there will be U, that will be our tRNA, U, C, J. So our tRNA will be U, C, J. So forming a new tRNA now, which is called U, C, J. So instead of forming our normal tRNA that we were forming before, remember the normal tRNA that we formed before was UCA. So the normal protein we were forming here was going to have UCA. That will be the normal protein. It will have UCA. But now the protein that we're forming now is having UCG. So meaning we're forming a different protein. So this is how we're going to answer it in the exam. Firstly, our normal DNA was what? TCA. Due to that change, we're going to form a new DNA of what? TCJ. And then the normal mRNA that we're forming was AGU. But due to the change, we're forming a new mRNA of what? AJC. Also, because we're forming a new mRNA of AJC, this also affects the new tRNA that will be there. So instead of forming UCA as a normal tRNA, we are forming a new tRNA of what? UCJ, which will lead to a different protein. That will be your answer for 4 marks now. Sharp. So 3.4.6, describe the process of transcription. Okay, I'm going to go through this quickly with this one. During transcription, we have a normal DNA strand at the beginning. The DNA strand will then unwind. Once the DNA strand unwinds, it will unzip, meaning we'll have 2 different strands. So the strand that we are forming, one will serve as a template. So the other one will serve as a template. So that template that will be formed there will be used to form an mRNA. So the template that has been formed will form what we call an mRNA. So using the free nucleotides which are there in the nucleoplasm, they're going to bind to that DNA template. And then from there onwards, you're going to form a complementary mRNA to the given DNA. Meaning if DNA was TCA, we're going to form an mRNA of what? AGU. If DNA was CJT, we'll form an mRNA of what? GCA. Because one strand will be used as a template. And for those ones who want a detailed or in point form, please check my playlist. I've done definitions of the whole transcription. So you can check my playlist there. Or you can just, again, I'll repeat again. During transcription, we'll have a normal DNA strand, which will be double helix. So the DNA strand will unwind. When it unwinds, it will break. And then those weak Abelian bonds are gonna break, which are holding the DNA together. And then now we're gonna have one strand which will be used as a template. So the one that will be used as a template, the free nucleotides which are there in the nucleoplasm will bind to that strand. When they bind to that strand, they will form a new mRNA. So meaning if the DNA strand that we're using as a template was TCA, the mRNA that will be formed there will be AGU, and then that's how the transcription will occur. So that will be worth 6 points for 6 marks. And again, for those ones who are new, please check our playlist because I've covered all these topics in theory first before I start doing these exam questions. And I hope this will be helpful to you guys. And I'm happy that I managed to finish the paper under 2.5 hours. And I still have 8 minutes to go and check my errors. I think it will be a good thing to finish early. So that will be that for this paper. And I hope that it will help you guys who are writing on Monday. Because I know that some provinces are writing on Monday, paper 2. So hopefully this will be a good help to you guys. Thank you very much.